Okay, today we're going to talk about how DNA replicates. So first there were three ideas as to how DNA might replicate, and we need to go through those first. So the first was the conservative method, and this is kind of like I've got a piece of paper and I need to get a copy of it. I go to the copy machine, I put my original down, and I get a copy, and I have my original and copy. That's called the conservative method, right? So I've got my original. When it's copied, I have my original and my copy, and then when I make copies of those, I have original, copy, and so forth. So that was the conservative method. But since DNA has this double helix, this double backbone, some people thought maybe there was this idea that a template was used. So this was called the semi-conservative method. And the semi-conservative model. So the idea here was that it sort of each of the helices served as a backbone, so that you ended up with the replicated DNA had half original and half newly synthesized DNA. And then when they replicated the same thing, each of those served as a template, and you had a semi-conservative with half of the original and half the newly replicated. And then the third was the dispersive method, and this was more like you took your DNA and just broke it up into a lot of pieces, and they all bonded with new nucleotides to produce the new DNA. So in this case, you'd have both helices would have both new and original DNA in them, and there would be sort of a mixture within all new DNA of the original DNA and the new DNA. So, experiment was done by Messelson and Stahl, where they said, okay, we need to figure out how this actually works, which of these three ideas or models is correct. So they looked at bacteria, and they knew that bacteria can replicate in about every 20 minutes, and they said, okay, bacteria, we're going to expose them for several days to an isotope, nitrogen-15, and they knew that would bind to the nitrogen bases, the A's, T's, G's, and C's in the DNA, and that all of those bases would have what's called heavy nitrogen. It's got that extra weight, that extra neutron in it. So when you centrifuge it, it's a little bit heavier. They then added regular nitrogen, nitrogen-14, in nucleotides that had the nitrogen-14, and allowed it to incubate for about 20 minutes. They knew that bacteria in ideal conditions will replicate all their DNA and divide in 20 minutes. So you'll have new sets of DNA within 20 minutes. And when they analyzed that DNA from the bacteria, what they found was that all of the bacteria basically had both the heavy and the light in it. It didn't have partially heavy and partially light. It was a mixture of both, so that it was either the semi-conservative or the dispersive method. Okay. Well, then they let that bacteria go for another 20 minutes, so that essentially this DNA could copy itself again. And when they did that, what they found out was that half of the DNA was the lighter form, half of it was a mixture of the heavy and the light, and that none of it was the truly heavy. So what this showed was that DNA was working this semi-conservative model. So our winner is the semi-conservative replication, because what they found was each of the templates, or the helices, was unzipping and being used to replicate, so that each new DNA molecule was half original and half newly replicated. And so it looks a little bit like this. So the blue double helix is the original DNA. It unzips, and we have half of the original DNA and half of the newly synthesized DNA on there. Okay, so some important enzymes are involved in DNA replication, and we need to go through those right now. The first one is we need to unzip our DNA in order to replicate it in this semi-conservative fashion. So the enzyme that unzips the DNA helices is called helicase. So again, helicase just goes through and breaks these really weak hydrogen bonds between the nitrogen bases and unzips the DNA. Now, if you've ever tried to separate small twines that make up a bigger piece of twine, like sometimes rope is made up of smaller pieces of rope, and that rope is coiled a lot like DNA. When you pull them apart, you find that it binds up. What happens is it just kind of doesn't work very well, and I'll try to demonstrate this in class to give you a good idea of how that works. So that's a problem when you're trying to replicate DNA in your bacteria. Every 20 minutes you've got to unzip and copy this DNA. So there's an enzyme called topoisomerase, and what this does is it's basically ahead of the helicase, and it's helping to kind of swivel the DNA and then even cut the DNA and let the DNA kind of move around so that it can continue to be unzipped without hindering this process, without preventing the DNA from getting unzipped. So that's an enzyme that kind of works upstream from the helicase and kind of makes sure the DNA can be separated into two helices by helicase. Then we have primase. This is actually a piece of RNA, an RNA small polymer, and it binds to the original site and kind of primes it. And basically, primer is a kind of paint, and when you put primer on like a rough surface, and it primes that surface, allows the final paint to bind to that surface. And that's kind of what primase does. It sort of binds to that original set of nucleotides and then allows the final kind of copy or the final pairing, which is done by DNA polymerase. So DNA polymerase then reads and pairs complementary nucleotides to each one of those bases and creates the new strand, these orange strands that you see right there. Okay. Then when we're done, we've got to kind of bind or glue together some of these new segments, and the enzyme that does that is called ligase, and lig is like ligament. Ligaments bind bone to bone. Ligase just basically glues pieces of DNA to other pieces of DNA in this process. So now let's look at some of this in a little more detail. So first off, here's our DNA helicase, and it's unzipping the DNA in that direction. And of course, again, because DNA is in this spiral configuration, it's going to bind up. And we have the topoisomerase here, which is actually spinning our DNA and kind of helping to unravel it so that it can be easily separated. And it even will go in there and actually cut the DNA and allow it to sort of swivel and unspin, unravel, so that it can be split by helicase as well. So there's topoisomerase and DNA helicase beginning that process of unzipping those two helices for replication. So the next step we have, now we've unzipped our DNA, is primase. And primase is going to basically bind to our DNA, and it's going to basically prime it such that it can then accept and bind to the DNA polymerase. And that's basically all you have to know about primase. So then DNA polymerase comes in, and it starts to, first off, it'll remove the RNA primer, and then it will pair up and match complementary bases. So A's to C's, A's to T's, G's to C's, and so forth, until we have two strands on there. Okay, and you'll notice here the DNA's got sides. It's got a 3 over here and a 5 over there, and 5s and 3s. What's going on with that? We need to talk a little bit about that. DNA is actually what we call anti-parallel, and the best way to think about it is DNA is more like a 2-lane road where cars go in one direction on one side of the road and in the other direction on the other side of the road. So these 3 and 5 prime sides are actually named for the carbons on the deoxyribose sugar. So the first carbon that binds to the nitrogen base is 1, the second one is 2, the one that binds to this phosphate is called 3, the one up here is 4, and then the last carbon that binds to the phosphate is number 5. So this is what's called the 5 prime side. Oops, go away. Because that phosphate binds to the 5. This is called the 3 prime side because it's bound to the 3 carbon over here. So you can see over here we're going in the 3 to 5 prime direction. Over here you can see the sugars are actually upside down. So the 3 prime side is over here, the 5 prime is over here. So this is the 3 prime side, and that's the 5 prime side. Now this is important for DNA replication because DNA can only be copied by DNA polymerase and replicated going in the 3 to 5 prime direction, which means DNA polymerase is going to go this direction on this side, and it's going to go this direction on this side. Okay? Well, that gets a little tricky because if we're unzipping our DNA, over here we've got the 3 prime side. Oops. And the RNA primer can bind there, and our DNA polymerase can move in this direction and basically continue to replicate, no problem. But over here we have a problem because we have the 5 prime side. That's the sort of way it's going to work against. It's got to start over here on the 3' side. Well, it can only start up here on the 3' side, and eventually it's going to run out of space, and it's going to have to double back as it's getting unzipped, and then it's got to double back and keep going. And when it does that, it's going to take a lot longer, and it's going to form these fragments. This is called the lagging strand. It's the lagging strand because it's lagging behind. It's going to have to keep doubling back and replicate a little and double back as the DNA gets unzipped. Okay? Those fragments that are produced are called the Okazaki fragments, discovered by this guy, Japanese guy, Okazawa. Go figure. Anyway, so that's why we have a lagging strand and a leading strand on DNA when it gets replicated. And then we have these pieces on here that are formed, and on the lagging strand, they're going to have to get sort of glued together, covalently bonded together by ligase, that last enzyme, which helps kind of seal the DNA by covalently bonding all those Okazawa fragments. So again, just to review, helicase unzips the DNA. That topoisomerase kind of helps to prevent the DNA from binding up as it's getting split. And then we have primase primes the initial site so that DNA polymerase can bind to it and basically undergo this process called elongation, which basically means pairing new nucleotides to the existing nucleotides. And then it also removes the primer. The DNA polymerase does that. The last step is that ligase has to seal up on the lagging strand all those Okazawa fragments, all those little pieces that were done, because it had to keep looping back. So that's the last enzyme and its role in there. Okay, so again, this is just an overview of that process. So DNA is unzipped by helicase. It always has to move from the 3' to the 5'. Notice the newly synthesized DNA is going to be produced in the 5' to 3', again, because DNA is anti-parallel. Okay, and then on this side you can see the fragments, the Okazawa fragments, are formed on the lagging strand, and they're glued together by this enzyme called DNA ligase. So this is just an overview of how DNA replicates itself. And in our next video, we'll talk about how DNA is copied into RNA in the process of protein synthesis.